Good afternoon, everyone, and welcome. For those I haven't yet met, I am Suwan Ko, Chair of the Cardinal and Gray Society and a member of the MIT class of 1973. I'm delighted to welcome you to today's Cardinal and Gray Society lecture on behalf of both the Cardinal and Gray Society and the Emma Rogers Society, and also on behalf of my husband, Victor Tong, a proud alumnus of the undergraduate class of 1972 and the graduate classes of 1973 and 81. It's been an honor to serve as your chair over the past few years. One of the great joys has been moments like this, bringing our community together around ideas that showcase MIT's imagination and influence. Before we begin, a quick note about tech reunions, taking place May 28th through May 31st. For those not attending in person, Cardinal and Gray members are invited to participate online, no matter your graduation year. Several programs will be live streamed, including an annual Cardinal and Gray Society Brunch and Technology Day, featuring MIT's new Generative AI Impact Consortium, known as MAGIC, and its quantum initiative, QMIT. Registration details will be shared next month. Now, today's lecture explores how emerging advances in biological engineering could help transform carbon dioxide conversion, resource recovery, and sustainable manufacturing. This work is increasingly vital to the future of our planet and our society. It is my great pleasure to introduce our speaker, Ariel First, Associate Professor of Chemical Engineering at MIT. Professor First's research sits at the intersection of chemistry and biomolecular engineering. She's advancing new ways to engineer biological processes that address challenges in environmental stability and human health. Beyond her research and teaching, she's committed to expanding access to STEM education and broadening participation in engineering, helping to shape a more inclusive and innovative future. Professor First, we're grateful to have you with us today and for sharing your work with our community. Please join me in welcoming Associate Professor Ariel First.
Thank you so much for the kind introduction and for the invitation to be here. I'm very excited to share some of the work that my lab does. Let me get my slides started. Excellent. Let me just get my pointer going. Okay, so I'm very excited to tell you about some of our work in how we can think about taking native biological processes and engineering them for sustainable technologies. Before I get into the specifics of the work we do, I need to thank my team and all the students that have done this work. Obviously, I'm not the one actually in the lab making these discoveries, that's them. I also started at MIT as a faculty member in 2019, which, if we all remember, was right before all of the COVID shutdowns. So many of my first students joined my group without ever having met me in person. And so I need to thank them for that leap of faith, and you'll see all of the amazing work they did. You probably also noticed that I have a very large number of undergrads in our lab. We work with a significant number of undergrads and even high school students, and they've made exceptional contributions to the work I'm going to talk about. So I will try to highlight their contributions, but I just wanted to make sure that I acknowledge them. So before I get into the science, I figured I'd give you a bit about my background because nobody in my family is a scientist or engineer. So my first introduction to research was actually as a high school student. I had the opportunity to participate in a summer research program where I got to do research in an academic lab, and my project was powering cell phones with vodka, basically. That's how it was sold to me, and what high schooler wouldn't be interested in that? In reality, what we were building was a series of enzymes, so basically just protein catalysts, to see if we could actually power things using ethanol as a power source. And because of kind of how freeing I found research compared to kind of conventional coursework, I continued research. I attended undergrad at the University of Chicago and worked in a lab that basically built proteins using chemistry. So again, kind of thinking about the interface of different disciplines in science. In grad school, I went to Caltech on the other coast, and I worked in a lab that looked at DNA as more than just the genetic code and looked at how it could actually be used as a wire. And it was in my grad experience that I realized, I spent 5 years building a colorectal cancer diagnostic, and I realized that it was very effective, but it was also very complicated. And so nobody was going to be able to use it. And so moving forward from there, I really tried to focus on kind of design-centered thinking and how we can actually build things that can be used by the people who need them. So in my postdoc, I went to Berkeley and worked on kind of environmental sensors that were disposable and easier to use. And that's also why I moved from my chemistry background into the chemical engineering department at MIT for my faculty position. So I just wanted to kind of situate everybody together in kind of how we think about biotechnology broadly before I get into the specifics. So we see biotechnology as where chemistry, engineering, and biology intersect. And specifically, I'm going to focus on molecular. So these are things like DNA and proteins, and microbial techniques to make useful products. So this is obviously a broader, biotechnology is a broader set of technologies, but we're just going to focus on molecular and microbial. And if we all remember the General Institute requirement bio class, the central dogma of biology is that we start with DNA. That is then transcribed into RNA, which is translated into a polypeptide, which is basically just a fancy word for an unfolded protein. And then we consider it a protein when it's folded. So basically, this is just to remind everyone that we can actually engineer this part by changing the DNA sequence. And when we think about how to do that in microbes, there are two different ways we can do that. We can either directly edit the genome. So this is going to be permanently integrated into the actual genome of an organism. Or we can add an external piece of DNA, a little circular piece of DNA called a plasmid, that allows us to control processes kind of outside of the genome. And this is transmitted between generations, but isn't inherently programmed into the genome. So for genome engineering, these are tools such as the CRISPR-Cas system, and they basically make these permanent changes. In contrast, we can make plasmids kind of outside of the cell and then put them into the cell and control them externally from the genome. So this allows us to have control over microbial processes while not having to go as far as genome engineering. And so when we think about all the potential uses of biotechnology, the ones in bold here are some of the things my lab specifically works on, but I'm sure everyone is familiar with kind of agricultural applications. I'll talk a little bit about biofertilizers today. For environment and environmental remediation, studying soil health and removing bad things. In industrial processes, we think a lot about how to make biopolymers and biofuels using things like carbon dioxide as a feedstock. And then in medicine, we think a lot about how we can move away from small molecule therapeutics, so small molecule drugs, and move towards things like whole microbes that have much more sophisticated processes as treatments. So I like to kind of describe how we think about science using a couple of quotes. I really like this one from Albert Einstein. We cannot solve our problems with the same thinking we use to create them. That's really why we use kind of bio-inspired design in our systems. And then similarly, Niels Bohr said, every valuable human being must be a radical and a rebel, for what he must aim at is to make things better than they are. That really is our goal with equitable technologies. And then lastly, I just like to show this one from Charles Darwin because I feel like it's very relatable. This was actually in his scientific journals that he kept records for when he was in the Galapagos. And he says, but I'm very poorly today. and very stupid and hate everybody and everything. And I feel like there are days when we can all relate, especially in research. So, how do we actually approach design? Well, many things that we think of as humans in kind of the built environment are actually inspired by natural systems. So this suit of armor here is based on the scales of the pangolin, and this is from the 1500s. This material here is swimsuit material from the 2010 Olympics that has since been banned because it gives swimmers too much of an advantage. And it is based on the structure of shark skin. And then this is just 3D printed material that mimics it. And then this last one here is Velcro, and you can see the kind of hook and loop structure. And I would say, most people think of Velcro especially as kind of a human invention, but it's actually based on is the coating of seeds that have these hook structures that can interact with animal fur, and that allows those seeds to be transported. So even when we think of processes that are kind of conventionally human derived, many of them are often inspired by natural systems. And so what my lab does is we try to take inspiration from those biological systems to engineer equitable and sustainable technologies. And so we do this across 3 main areas. In human health, we make diagnostics that are inexpensive. We try to make models that allow us to study pathogenesis. And we make things like microbial probiotics to replace small molecule medicines. In environmental remediation, we do things like build sensors for monitoring soil health. We build systems to degrade contaminants. And we see if we can actually take valuable resources from what is generally considered waste. And finally, in the sustainability area, we think about how we can use natural metabolic processes of microbes for things like bioenergy production, carbon dioxide capture, and critical resource recovery. So I'm going to start with a human health story. So I'm not sure how familiar everyone here is with this, but this was kind of news when we had started this project to me, which is the majority of cervical cancer, about 98%, is actually caused by a virus, HPV. And there's a significant difference in the mortality rate from cervical cancer, and about 90% of mortalities from this cancer are due to lack of access to healthcare. And so this is my postdoc Marjan, who started this project in my lab, and my first graduate student, Cindy. And what they were inspired to do was to think about how they could decrease the cost of a diagnostic system and make it easier to access. And so Marjan was really motivated by gold leaf. This is the same material that you get in fancy restaurants on your food. And she was thinking that if it's cheap enough to put on food, it's probably cheap enough to use as an inexpensive electrode material. And so that's exactly what she did. She built these kind of flexible electrode systems that have about 16 cents worth of gold in each electrode. And this whole device is about 50 cents. And so I'm not going to go through each of the individual steps here, but I just kind of want to show you what this workflow is that we can do solely on chip now. If we have a viral or bacterial target, in this case, high-risk HPV, we initially do isothermal amplification, that's just single temperature amplification. We then use that amplified material to activate a protein, in this case, a Cas12. If that protein is activated, it will then cut DNA that's on our electrode, turning our signal off. So basically, amplification, and then we get specificity with this activation because we have to have the proper complement to guide RNA that's loaded into that protein. And then once we actually apply it, it's amplification again because that each protein will cut more than one DNA strand. And we can see what this looks like for high-risk HPV here. These were cervical swab samples blinded from patients. And we can see that basically this is the limit of detection with existing approaches. And we see that we get basically a sensitivity, and 98% selectivity. We have one false positive here, but otherwise this is an exceptionally sensitive technique, and we can get down to detection of 100 copies of that virus, so very low detection limits. And if you notice here, really all we're doing to detect HPV specifically is changing the sequence that we use to amplify the DNA and changing the guide RNA. So because it's so simple to kind of change this, we've been able to develop sensors very rapidly for all of these different targets just based on swapping out those sequences of DNA. And I think one of the really key advantages of this is that electrochemical detection is actually really straightforward. So we've been able to develop these very small potentiostats. They're about the width of 2 quarters. And these cost us about $25 to $50 each, depending on where we order from. We can integrate that with this heating system that allows us to control the temperature to actually run those reactions I was talking about. And this whole system is basically handheld size and shape. So this is fantastic because now we have this diagnostic where we can really change out what we want to sense by just altering what DNA sequences we throw in for amplification and readout. The problem is, if you notice here, this still requires a battery pack. And so kind of the next step for us was to move this to colorimetric detection. And so a postdoc in my lab was able to develop these kind of single bipolar electrodes where now when you have your DNA that has this probe on it, if that DNA remains intact, it will trigger a color change at the other end of the electrode. And if that DNA is cut, we no longer have this redox probe there and we no longer see the signal. So you can see the color change here. In the middle of this electrode system, this dark purple. So it's a very strong change in color. And if we use that same assay that I mentioned before, where we have this activated protein, we can see that if we have RNA that is the proper sequence, we basically see our signal turn off. Whereas if we don't have that, our signal remains on and we maintain a color. And we can actually get quantitation here by just looking at the RGB values, which are just the color change values in those 3 channels, using a simple cell phone picture combined with some automated software. This device can actually be powered by a watch battery, and so this is even simpler because it requires no hardware or smartphone in order to connect. So in this kind of first story, what I hope you can kind of see is when we are developing these assays, we try to kind of avoid issues with each individual assay type. So if you notice, we integrated kind of amplification, which decreases our detection limit, with specificity through biomolecular interactions. So we have to have that DNA interacting with that guide RNA in order to turn our protein on for sensing. We also wanted to ensure that the people who needed these sensors could actually use them. So we've developed multiple form factors that both use and don't require things like a smartphone or external power pack, so that based on the resources at a particular location, we can kind of tune the use and the need for these systems. And this is fantastic for diagnostics, but something we want to think about is a little upstream from there. Can we actually prevent exposures that cause these adverse effects? And so we're going to change directions a little bit to environmental remediation. But one key area of interest for me is kind of agriculture and lack of safety there. And so these are called organophosphate pesticides, and they're about 50% of the pesticides used globally. And if you notice, they are very chemically similar to these compounds up here, which are chemical warfare agents. And one of the challenges is that most agricultural workers distributing these pesticides don't have this level of personal protective equipment. What this means is there are about 10,000 deaths annually due to exposure to these pesticides, and that low-dose long-term exposure can lead to everything from higher rates of infertility to cancer. So what we wanted to do is to give these workers a way to both monitor their exposure levels and prevent inhalation, which is the main route of exposure and neurotoxicity. The great thing is nature had already evolved a protein that is very good at degrading these compounds. Organophosphate hydrolase, or OPH, was identified about 3 decades ago, and it basically cleaves this organophosphate pesticide into 2 benign molecules. You generate a phosphate, and you generate this p-nitrophenol, or as I'll refer to it, PNP. And with this PNP, it's actually a yellow color, so the way most people detect it now is just with UV-vis absorbance. The challenge with organophosphate hydrolase, and the reason we don't have better protection using it, is that it's a very unstable protein. So as soon as you express it and isolate it and then try to turn it into a material by scaffolding it on a resin or on a polymer, it unfolds and loses its activity. So but the first postdoc in my group, Amrita Karbolkar, and one of our first undergrads, Rachel Almark, who started with us as a freshman and stayed all 4 years with us, what they thought of was, how can we both decrease the cost of using proteins while also improving the stability? And what they found was this surface display approach. So if we just add a short amino acid sequence to this protein, it tells the cell to put it on the surface. We've also found conditions where we can freeze dry these cells, so basically turn them into astronaut ice cream, and the proteins remain active, but the cells are no longer alive, so we don't have to feed them. And when we use this approach, you can see here that with 0.1 weight% of these cells compared to the pesticides, so when you think about the whole cell mass compared to the pesticide mass, that's even a lower amount of actual catalyst of the organophosphate hydrolase than you would normally use. And that was really the only way we could actually get kinetics out of this data, because this reaction occurs so quickly because we've stabilized those proteins on the surface. So what I'm showing on the right here is just UV-vis detection of this small molecule. So this is that p-nitrophenol, that yellow molecule that's generated after you cleave the organophosphate pesticide. And so this is the concentration of our pesticide. You can see in all cases, we basically get 100% degradation, so full generation of that p-nitrophenol. The problem here, if anybody here remembers their analytical chemistry classes from undergrad, is we see very large errors at both 50 micromolar and 25 micromolar concentrations. And so when we back calculate the limit of detection from this amount of error, we actually get concentrations as high as 50 micromolar for our consistent limit of detection. And 50 micromolar is higher than the lethal dose of this pesticide. And I would argue that a sensor that only detects at above the lethal dose is not an especially useful sensor. So our second question, so now that we have these materials that can degrade this effectively, how do we actually sensitively and reproducibly detect the degradation products? And so we turned to biology. We were very lucky and we found a transcriptional regulator, which is just a protein that turns on and off genes, that natively binds to small molecules that look a lot like our p-nitrophenol. So when we put green fluorescent protein under control of this transcriptional regulator, you can see that with the small molecule, as you would expect, we see a lot of green fluorescence. The challenge here is even without this p-nitrophenol, we still see green fluorescence because of leaky or nonspecific expression. And so that's unfortunately not going to be a viable sensor, because we won't know if our on signal is due to a low concentration of that molecule or because of this kind of background expression. So what we did instead was to put this GMPR protein to control this protein SIM-A. And SIM-A is from a microbe called Shewanella onadensis, which was originally isolated from pond scum in upstate New York. And these microbes can actually spit out electrons. They have a really unique respiratory pathway that can spit out electrons extracellularly. And they do this in nature to reduce metal oxides, basically to free up those metal ions for other members of their community. In the lab, though, if they're fully intact, we get current generated on an electrode. So we basically have an electrochemical sensor. The great thing is these are genetically tractable. So the way the electrons get from the inside of the cell to the outside is through this pathway called the MTR pathway. And the first protein in that pathway is called SIM-A. So we were able to knock out this protein. So we basically cut the wire or unplug these cells, and we complemented it back in under control of this GMPR. And so now, as you can see, only when we have those engineered cells plus that small molecule do we get current generation. And normally, these Shewanella would not like to live in the same environments as E. coli. But if you remember, we've already freeze-dried our E. coli and killed them. So we can mix these enzyme materials with our Shewanella to basically get one-pot degradation and detection. And if we look at the right here, we see that this has very high specificity for both of those microbes plus the full pesticide. We do see a little bit of background expression, which we think is from the degradation of the pesticide through UV light exposure. What was even more surprising to us, though, was that this is actually quantitative. So as we increase the concentration of pesticide from 1 micromolar to 20 micromolar, we proportionally increase the total charge. And our limit of detection with this is now sub 1 micromolar. And so what that means is we've now lowered our limit of detection by almost 2 orders of magnitude, by almost 100x, by simply just moving from absorbance measurements to biological systems. And now we're also below the limit of detection, or excuse me, below the toxic limit of this compound. So our limit of detection is now well within a relevant range to prevent poor health outcomes. So that was great, but my grad student working on this project, Alec Aggie, and a postdoc in our lab, Tali Doughton, came to me and said, well, it's great that we have this transcriptional regulator for p-nitrophenol, but what about everything else we want to detect? We don't want to have to go digging to find a transcriptional regulator for each of these compounds. And so they've been able to basically generalize this platform using a native microbial process. So in microbes, the way they sense their environment natively is with these 2 component sensors. We have the first component, which is basically a protein that's usually membrane associated, that's either a transporter or a binder for your target molecule. When this protein changes conformation due to a binding or transport event, it basically starts a phosphorylation pathway that turns on or off a gene. This overall system sounds a lot like our standard Shigella sensor, right? So if we take a native phosphorylation pathway in those Shigella and hook that up to our sensing system, we found that we can effectively borrow this first part of these sensors from pretty much any microbe and effectively express them in our cells. And so far what we've done is focus on environmental contaminants as well as soil health indicators. They're all shown here, and they're all from different strains of microbes. So none of these are necessarily from that native Shigella strain. But we can detect all of these at or below the relevant concentration for contamination. Not only that, but oftentimes there are emerging contaminants of concern. These are often kind of things that are found to be harmful later on after they've been used extensively in things like manufacturing processes. And so Kruti in our lab, who joined as a sophomore in high school and has been working with us since, she's now finishing her freshman year at MIT as an undergrad, developed a program that allows us to search for these sensing systems in the Kyoto Encyclopedia of Genes and Genomes, or the KEGG database. So what she realized is that generally people have annotated enzymatic reactions, so catalytic reactions downstream, much more than they have these sensor systems. So what she did was look for kind of allowed reactions with enzymes for a target molecule. And then she backtracked to build candidate pathways and ID the proteins that are likely participating in these sensing systems. It also then pulls out the species that likely contain those pathways and tells us exactly in our genome where those sensing genes are. And so taken together, what this basically does is allow you to pop in a chemical and pop out where this program thinks that there will be basically sensing gene systems in a target organism. And if it sounds too good to be true, well, Kruti actually showed that it works really well. She focused on vinyl chloride, which is the contaminant that was spilled during the East Palestine, Ohio train crash a few years ago. And she was able to not only identify 9 novel sensors for vinyl chloride and similar compounds, but she was able to clone them into cells and actually validate that they were what she said they were for, just simply using this search system. And so our goal is to have this be kind of a broad tool for people in this realms to be able to identify new sensing systems to transform them into our sensor platform. So this is seeing a pattern that we very much like platform technologies in my group. The problem is when we actually think about how people would use these sensors, this is a picture of how they're actually constructed. We have this jam jar that's around 100 milliliters. We add cells to it, and then you can see our electrodes are in there. The problem is if we think about our initial targets, which are basically agriculture and industry, they don't want to have to set something as complex as this up in their system. So what my grad student Alec came up with is basically transforming this from a kind of more conventional liquid phase electrochemical system into an RFID tag. The challenge with RFID tags, which are basically just large or very small antenna, is that the time scale at which electrochemical processes operate is much, much slower, 3 orders of magnitude at least, 5 orders of magnitude lower than where these RFID tags operate. So how do we bridge the gap between the time scale of these biological processes and the time scale of the RFID operating device? And the way we do this is with a collector generator system. So if we have 2 electrodes that basically face each other, conventionally, if we throw in ferrocyanide and ferrocyanide, those 2 will cycle with each other, basically bouncing between these 2 electrodes and generating a significant amount of current that can actually power our RFID tag. In contrast, when we actually express our kind of sensor system, our sensor circuit in Shewanella, and it generates the sensor, we now have reduction of our target species in solution, basically through the microbe and not through the electrode. And so what we see here is a significant attenuation because basically we have now disrupted one of our surface processes. And so this basically turns off the power to our RFID tag. So if we look at how this functions with just our Shewanella, so just our microbes that are electroactive versus our Shewanella that are effectively unplugged. Remember, this is the knockout here. And you can see that our standard RFID sensor turns off immediately, while our knockout pretty much never fully turns off. And because we have such precise control based on the concentration of ferrocyanide in the device, combined with the time of detection, we can basically tune these in for a specific time scale of sensing based on having a less than 1% false negative rate and less than 5% false positive. And what you'll notice here is that this is at 10 ppm paraoxin. What we actually find is if we titrate in that paraoxin, our limit of detection is down at around 100 parts per billion. So that's an order of magnitude lower than what we observed with kind of the bulk electrolysis system. And what this actually looks like is it's a pretty simple setup. We have those gold leaf electrodes that I described in the first story in the talk, separated by a cellulose separator from a carbon paper electrode. We can just 3D print this half of a cap and put a sample well on top of that. What it looks like in practice is this, where this is our sampling system and this is specifically our kind of sample well. We have that connected to an RFID tag, which is also connected to an external switch and battery. And because these are all off-the-shelf components, it costs us less than $2 per device. And so what we're working on now is basically translating this approach into something that can be broadly deployed, even autonomously. Because RFID tags are so prevalent, there are free sensors for everything from cell phones to Apple Watches, and we can even use RFID sensors on things like autonomous drones or vehicles to monitor without having to have hands on in that situation. So I hope what I've shown you in kind of the second story is that we need to understand how our system works in order to develop our system as a technology. But in order to really solve this challenge of these organophosphate pesticides, we needed to both have a fundamental understanding and then be able to apply our lessons to support this worker health. If we think about kind of native sensors from other microbial species, our goal was to develop our initial technology as a platform where we can integrate components of pretty much whatever we want to develop new sensors for pretty much any target of interest. And finally, as I mentioned in kind of the first part, if you have a sensor, but nobody can use it because the form factor is incorrect, then no one will use your sensor. So we focus more than I think I would normally on improving the form factor to develop it as an RFID tag to enable the broad use of this tag. And so as you've probably picked up, we really like platform technologies in our lab. So we talked about how we can make that sensing platform into a platform technology from just a single transcriptional regulator. But I also want to emphasize how we can use the other part, these freeze-dried microbes with these surface displayed proteins, kind of as a platform tech. So I'm sure when many of you saw proteins in my talk, you said, oh, those are too expensive to use for most environmental applications. So this doesn't matter, right? People have been using enzymes for years in industrial processes that are highly optimized, such as the additives to food and animal feed, in detergents and cleaners, and in the chemical manufacture space. In fact, it's an $8.5 billion market currently. However, one of the challenges with deploying a new enzyme for a new process is the cost and expense of the downstream processing of these proteins. If we start out way back at the beginning of my talk, I talked about engineering these plasmids to express what we want. If we do that and put them in the cell, we can very readily grow that cell up in a large fermenter to generate a very high density of cells in that system. But what actually takes up the most kind of energy as well as cost is the downstream isolation, stabilization, and processing. And this accounts for as much as 90% of the cost of implementing a new protein. And so what we've been looking for is applications of these protein materials where, one, you get a more stable protein, and two, you can use this for both kind of environmental remediation, but also value-added products. And so what we see here is that if we think about enzymes, so things that can do catalysis, we have applications ranging from kind of pesticide degradation to artificial photosynthesis. In contrast, we can also kind of recover things with proteins using the specificity of those proteins for everything from rare earth element recovery from kind of complex mixtures or coal fly ash, all the way to heavy metal removal from water sources. And so I'm just going to focus on one of these applications beyond the pesticide degradation, which is rare earth elements. I'm sure everyone has heard of kind of the current discussion around rare earths and supply chain limitations with them. That's because rare earths are used pretty much ubiquitously in technology now, from everything from electronics and batteries to radar guidance systems. The problem is we actually have enough of these metals in the US to use them right now, but less than 1% are recycled because of the harsh conditions required for leaching and regeneration of these systems. And so what we thought we could do was use a protein that had been known for about a decade called lammodulin, and it's known to specifically bind to 3 rare earth elements. So we can see that here with these 3 balls. We see them in the structure here. And this is actually a pretty poor representation of the structure because without the rare earths bound, this is actually very floppy and very inconsistent in terms of binding. I will note this work was done by a postdoc in my lab, Dr. Melanie Good, as well as an undergraduate, Tatum Willem, who's now at Stanford for her PhD. And I'll talk more about the technoeconomics later, but basically these cost us around $10 a kilogram for dried cells, and we get about 12 milligrams of lanthanide per gram of cells recovered. And so what we've been able to do initially is just to develop this as a packed bed filter. So if we put these surface displayed protein materials in a filter and flow through our kind of mixture that contains rare earth elements, anything that is not specific for that protein, so things like magnesium, calcium, iron, all flow through, while we get our target molecules, our target lanthanides kind of bound to those materials, which we can then release, as you can see here, with just a lower pH wash. So shown across the course of 10 days, we see this cycling of binding and release. And what this shows us is that we get about an 83% total recovery. And that corresponds to about twice as much recovery as current methods for rare earth separation. I should also note that currently rare earth separation is performed with about 300 liquid-liquid separation steps that require chemicals such as kerosene and phosphate ligands that are known to be very cancer-causing, carcinogenic. And so what we see here is basically that we end up with these rare earths in water because we're not adding any organic solvent in order to bind or release them, and stable protein-based materials across 6 days without requiring cold chain. So now we have protein materials that we can use for separations that don't require the same consistent kind of temperature and controlled environment transport that you would normally associate with biologics. So where do we go from here with this? Well, as you noticed, that last one was just kind of a mix of lanthanides. So I walked into lab one day and my postdoc and one of her undergrad mentees were standing at her bench wearing big goggles and holding hammers over a bunch of light bulbs. They were actually able to crush up light bulbs and extract using acid the rare earth from that. When they loaded those onto our column system, what they saw was some specificity for particular rare earths based on the pH. And you can kind of see that with scandium and yttrium compared to these other lanthanides, where this kind of mid-range pH is what releases most of the metal compared to yttrium. And so our goal now is to engineer these proteins basically to give us more specificity based on pH for release, so that we continue to only have aqueous waste streams without generating any toxic chemical waste, and enabling both one-step binding and purification of individual rare earths. So as good engineers, let's think a little bit about the technoeconomics of this. If we think about how we conventionally make proteins, as I mentioned, we start out with plasmid engineering, and that is very inexpensive and well established. And once those cells have that plasmid, we can grow them up in fermenters very easily. The challenge comes in the downstream isolation, stabilization, and purification, which is about 90% of the cost. So with our surface display approach, we basically skip all of that. Oops, sorry. So to make these at scale, we require no specialized equipment, and it's less than 10% the cost of conventional proteins. What that means for us is that we are now able to offer a solution for people trying to extract rare earths either from waste streams or from very low concentration sources, not because it's the sustainable approach that's much better for kind of earth than kerosene-based separations, but we can do it because it's profitable for them. As we can see here, if we actually reuse those protein materials, it costs us about $150 a kilogram of rare earths. So even if we don't recycle those materials, based on our estimates, it's around $450 a kilogram. And the commodity pricing of these rare earths is around $3,000 to $5,000 a kilogram. So again, what we're emphasizing here is that the sustainable choice is also the profitable choice, which we anticipate is going to help with kind of commercial uptake of this type of tech. And so the last story I'd like to tell you today is just a short one on how we can use materials we developed in my group as kind of protectors of microbes that allow us to apply microbes in new and creative ways. I do want to disclose that I'm the co-founder of a startup called Seya Bio based on this work. And this work was done by many different people in the lab, including a former postdoc in the lab, Dr. Gong Fan, who's now faculty at Rochester, Grace Anderson, a grad student, Marjan Smani, postdoc, Ben Burke, grad student and undergrad from ChemE at MIT, and Yukyung Go, who's a materials scientist. And basically what we've been able to do is to take materials that self-assemble and use them to form these two-dimensional coatings on the outside of microbes. These coatings are basically, are made from food-grade components, polyphenols. These are things like tannins or green tea extract, combined with earth-abundant metal cations. So these are things that you'd find in your multivitamin, like iron or manganese. And when we mix those together, as long as there's a nucleation site, so something for that coating to start forming on, they form these two-dimensional networks very rapidly and readily, which we can see in this video. So if you look on the left, that blue color forming is us actually coating microbes. As soon as that metal chelates to the polyphenol, we see ligand to metal charge transfer, or the color change that results from their mixing. And the nice thing about this project is based on the change in the metal and or the polyphenol, we get this beautiful rainbow of different colored materials. The other great thing about this tech is we find that it coats each individual microbe uniformly. We can see that by transmission electron microscopy here, while also coating a full batch of microbes equivalently. So you can see the core shell structure of the microbe coated in these materials here across this whole batch of microbes. So each individual microbe is fully coated, and every microbe in the batch is coated. And so then I tasked my students with basically thinking about every sort of kind of stressor they could think of in the lab to expose microbes to. What they came up with is oxidative stress, including UV irradiation, as well as the addition of things like hydrogen peroxide to our cell culture. Mechanical stress, which is often associated with spray drying or freeze drying processes, chemical exposure, radiation exposure, temperature fluctuations, both hot and cold, high humidity, as well as high salinity. And what we find is that across all applications we are interested in, these coatings protect each microbial strain from stressors of interest. So this ranges from agriculture, where we can do things like replace fertilizers with microbes. In human health, we can make vaccines that don't require cold chain or make better probiotics to help your gut. In green chemical production, we can make these microbes more tolerant to high levels of either solvent or the chemical product in solution, making us able to basically make more product per batch because we can go to higher titers of that target chemical. And in the built environment, we can use microbes for everything from metal recovery from e-waste to the generation of self-healing building materials. When we think about the scalability of microbial tech, because these coatings self-assemble and they're all kind of food grade commodity chemicals, we can make these microbes for basically $10 a kilogram. We can apply these coatings in media in our fermenter, and we end up with as many as 1 million times more live cells per batch than we do with uncoated cells. Because all of the coating components are generally recognized as safe by the FDA and are available at commodity scale, it's a very inexpensive technology to implement that really only has benefits. So to give you an example, if you were to buy a year's supply of probiotics, so every pill you would take for a year, the additional cost of coating, not even adjusting for the number of increased microbial, or sorry, the increased number of microbes in each batch, we only add an incremental cost of around 25 cents. So even without accounting for the increase in yield, it still only costs 25 cents to add this tack. And what we can do with this is protect against everything from very high heat. So what's shown here, is the tuberculosis vaccine, which is just a microbe. This is also used as a bladder cancer treatment. And we can see that if we dry down these microbes, we can store them at 50 degrees Celsius, that's around 130 degrees Fahrenheit, for at least 3 weeks. That's this light blue bar. And all we're looking at here is just changing the number of coatings. So as we increase the coatings, we also kind of increase the stability here. And so that gives us more protection against stressors. What this also means is that the places where the tuberculosis vaccine are needed the most, including in developing nations, no longer require cold chain transport or storage in order to maintain supplies of this vaccine. Similarly, if we think about kind of how we can use these microbes for improving construction, this was actually a surprise to me, but around 9% of global CO2 emissions are specifically from the construction industry. And a lot of that is actually from transportation of materials and hands-on need to fix things. And so all we did was dry mix in some of our microbes kind of coated in these protective materials. And when we do that at a 0.1 weight percent, in a month, we see about 35% crack healing. You can see that in these images. So the live cells here, you can see kind of the white filling in here, versus the dead cells, we see very little to no kind of healing there. And what this also means is that you now have a building material that can basically do direct air capture. So the way these microbes actually do self-healing cement is by taking CO2 from the atmosphere and attaching it to a calcium or magnesium ion to basically make limestone. And we can show that the improvement we get with just having our coated live cells in this dry mix enables significant crack healing. So these are about 0.5 centimeter wide cracks. And we get about 40% of that crack filled in. And because we get very little background filling with those dead cells, what this tells us is that having these cells alive and happy in this matrix is critical for the success of this. I also want to note that these are 10 centimeter by 10 centimeter cement pucks. So these are actually fairly large kind of cracks that have formed and that we actually see visible changes in healing for. I also want to note that using this protective approach, we can take microbes that generally aren't stable enough to be used in bioreactors and use them in those systems. So I'm sure you've all been asking yourselves since I showed that original slide of my group, why they were all dressed like cows. And one of the reasons is because we use methanogens, which are microbes that can take CO2 and produce methane. And one of the strains we use has been isolated from the cow gut. So what we want to do with these microbes is enable them to basically generate fuel materials from CO2, which would allow us to circularize the carbon economy even for kind of tough to decarbonize industries like aviation. And so conventionally, these microbes take hydrogen and carbon dioxide and turn it into methane. The problem is this hydrogen is very energy intensive to produce and can be very precious. So what we've been able to do is swap out this hydrogen for an electrode and see that these microbes, especially when they're coated and protected, actually convert this carbon dioxide to methane with very high efficiency at about 30% lower energy than is required to generate your hydrogen for conventional. And finally, I'll talk about kind of the impact on agriculture. So microbes broadly are known to support the majority of life on Earth, including plants. They cycle nutrients in the soil, they can supplement nitrogen and phosphorus, and they can even secrete pesticides on site. The problem is most of the microbes in soil are very happy in their community of diverse microbes. And so when we remove them from that and try to scale up their production, they all die because they don't have their other community members around. Additionally, many of these microbes are pretty temperature sensitive because they're used to being kind of buried in the soil and also away from sunlight. And what we can see here is if we coat one strain of these microbes called Pseudomonas fluorescens, which is both a pesticide secretor and a nitrogen fixer, if we heat these guys up to 50 degrees Celsius for a couple weeks, we see that all of the coated survive while most of the uncoated don't survive. And so this is why these couldn't be formulated previously for agricultural use. What was even more surprising to us than just the difference in microbial survival, though, is that when we apply these to seeds during planting, what we get is a germination rate that's about 150% higher than even the fresh microbes. So our coated microbes that have been dried and stressed are even more effective than fresh, which is really one of the key limitations in regenerative ag right now is the need for growth of your microbes on site. And so what we've been able to find with these kind of agriculturally relevant microbes is we no longer need liquid transport and storage and cold chain storage, which saves a significant cost and energy input in terms of shipping and transportation of these materials. We no longer have to transport these materials as hazardous waste or hazardous materials, or farmers don't have to receive things like ammonia and urea, which are hazardous materials to transport and store. Because most of these microbes can be delivered at the time of planting, you also save a significant cost in terms of equipment and labor from not having to refertilize mid-growing season. And because many of these microbes can also secrete pesticides, and they generally do that only in the presence of that pest, so whether it's a fungus or an insect, which means you're significantly decreasing the environmental impact of farming because you're decreasing the use of these toxic chemicals. And you're also saving costs to the farmers by not requiring them to actually purchase those separate chemicals now. So I hope what I've showed you with kind of this last story is basically that as we think about making technologies that allow us to bring the cost of biological systems down, we can start to think about applications of these biological technologies that would normally be cost prohibitive. And that's both for these whole microbes as well as for our use of enzymes with surface expression. And overall, I hope what I've showed you in the seminar is just that, as we think about applying biological insights to technologies, we can make things that are both cost effective and sustainable. And apologies, I think I went a little bit over time, but thank you for your attention and I'm happy to take questions.
I don't think we have too much time to have questions, but it was such an interesting talk. And I just want to, there was a couple of questions that I would have asked is like, what role can the institute like MIT and its alumni play in accelerating this kind of innovation? I think we could end with that
Excellent.
question.
Um, Well, one, I think I feel every day here that I am standing on the shoulders of giants. So thank you to all of the alumni for all of your contributions, kind of beforehand. And, everything from introductions to, I will send out the link for Say a Bio. If you want a sample of microbes, we're trying to just get feedback on it now for home gardeners. So if you want to try them in your home garden as you start planting for summer, just drop us a line and we're happy to send you. I know I went over time, so I'm happy to answer any questions that people have that didn't get answered by email. My email, I'll make sure it gets sent out afterwards. So, yes.
That was wonderful. Thank you so much. It's just so many areas that's been touched. And it's like, oh, our life is going to be sampling some of these things. So thank you
Excellent.
very
Yes.
much, Ray. And I
Thank you.
wanted to put one more plug in to our tech reunion coming up. You all are welcome. As I said, Cardinal and Gray, we're always there. Thank you so much.